Integration of metabolism. In this slide presentation, we will continue to review the metabolic processes dominant during different metabolic environments. We first have discussed the metabolic processes dominant in a fed state, and also those processes dominant in a post-absorptive or early fasting state. Next, we will talk about the processes that are occurring at a higher rate during a more prolonged fasting state and during starvation. As a reminder, a fasting state is occurring 18 hours post-meal intake up to 2 days after consuming food. We will talk about the processes dominant during this fasting state, more prolonged fasting state. So we can see that gluconeogenesis is going to be one of the primary processes, and this process of gluconeogenesis is going to involve breaking down of muscle protein and converting particularly glucogenic amino acids to glucose, and the glucose is going to be used to supply blood glucose levels, which have declined rapidly during this more prolonged fasting state. Additionally, in this metabolic environment, we're going to see increased rate of beta oxidation, or the oxidation of fatty acids to produce ATP, and we will also begin to see the formation of ketone bodies through the process of ketogenesis. So while during this more prolonged fasting state, we're going to see a greater increase in the process of gluconeogenesis to supply glucose to the blood, we're going to see a difference now between the fasting and more prolonged starvation state. So this is going to be beyond 2 days without food intake, and we're going to see a shift from gluconeogenesis to beta oxidation. So beta oxidation at this point is going to be dominant. Also dominant during this more prolonged starvation state, so without food intake for 2 days or beyond. We're also going to see a greater increased rate of ketogenesis. So at this point, both the beta oxidation and ketogenesis is going to be dominant. Ketogenesis is going to be dominant primarily after we see depletion of the intermediate oxaloacetate. And as we saw previously with the slide presentation on ketogenesis, as oxaloacetate levels decline, as they are used to supply, to be used as a substrate for gluconeogenesis to supply blood glucose, as oxaloacetate goes down, that decreases the activity of the Krebs cycle, because at that point oxaloacetate cannot be joined with acetyl-CoA to form citrate. So as oxaloacetate declines, Krebs cycle activity goes down, and then acetyl-CoA levels go up, and the acetyl-CoA is going to be used for the formation of ketone bodies. Since at this point, glycogen levels in the liver and the muscle have long been depleted, and also we see a decrease in activity of gluconeogenesis because substrates used to undergo gluconeogenesis are also declining. We now see that a major source of glucose is the glycerol backbone that's going to be freed during the process of beta oxidation. So again, at this point we see beta oxidation being dominant, freeing the free fatty acids to form acetyl-CoA, and then also with beta oxidation we have freeing of the glycerol backbone, which again at this point is going to be a major source of glucose. So we see in this diagram the activities occurring during a prolonged fasting and particularly starvation state, and we see these processes occurring in the liver, in the adipose tissue, and in the muscle. So we'll focus on these 3 tissue sites, as well as the processes dominant in a prolonged fasting and starvation state. So first, when looking at the muscle, we see the process of lipolysis, or the breakdown or hydrolysis of the triacylglycerol molecules to the free fatty acids and the glycerol backbone. So we see with the breakdown of the triacylglycerols, we see the fatty acids being freed. The fatty acids will then be picked up by the liver, and then in the liver we see the process of ketogenesis, so the fatty acids being converted to ketone bodies. Also we see with the breakdown of triacylglycerol, the freeing of the glycerol backbone. The glycerol backbone will be taken up by the liver and be converted to glucose through gluconeogenesis. When looking at the muscle, we saw that previously in the muscle we see breakdown of muscle protein, so this is going to be in prolonged fasting and starvation state. we see less of the alanine glucose cycle and more of the actual breakdown of muscle protein to free amino acids. And so those amino acids will leave the muscle and be circulating in the bloodstream, be picked up by the liver, and undergo gluconeogenesis, as well as lactate formed in the muscle will be picked up by the liver and also be used to form glucose. So again, we see the key metabolic processes occurring in this prolonged fasting or starvation state. We're going to see oxidation of fatty acids through the process of beta oxidation. This is going to be occurring in the liver. We're also going to see beta oxidation occurring in the muscle. We're going to see ketogenesis. So that's going to be occurring in the liver, so the conversion of acetyl-CoA to ketone bodies. Additionally, we're going to see the process of gluconeogenesis, with the gluconeogenic precursors being the amino acids freed from the breakdown of muscle protein. So we're going to see the breakdown of muscle protein, freeing the amino acids. We're going to see the conversion of glucose to lactate, and also the glycerol backbone. That's going to be freed in the hydrolysis of triacylglycerol. So those three substrates are also going to undergo gluconeogenesis in the liver. So now we're going to discuss the regulation of metabolism and the key signals that will be circulating in a fed, fasted, or starvation state that will be promoting those metabolic processes that we just discussed. So first we're going to discuss the signals that will be elevated in a fed state, and these signals in part will be involved in activating key enzymes involved in anabolic processes. So in the fed state, key hormones or other signals that will be elevated will be those related to the process of digestion. So we previously talked about secretion of gastrin, secretin, cholecystokinin, and also we see secretion of G1P. So these will be hormones that will be circulating in the fed state as a result of digestion. And then post-digestion, we're going to see increased circulation of insulin, and that's going to be in response to increasing blood glucose. A hormone that is going to be representative of a more long-term indication of adequate energy status will be leptin. And also we'll see that the malonyl-CoA is going to be elevated, and this is going to be in result of a higher ATP versus ADP ratio, so more ATP compared to ADP. So with the higher ATP compared to ADP ratio, we'll see conversion of acetyl-CoA to malonyl-CoA. So these are going to be key hormones or other signals elevated in a fed state. These hormones or other signals, and in particular insulin, is going to be involved in dephosphorylating these key enzymes during a fed state. So these key enzymes will be dephosphorylated. So glycogen synthase and acetyl-CoA carboxylase will be dephosphorylated, and that is going to activate those enzymes. Phosphorylase and triacylglycerol lipase, on the other hand, will also be dephosphorylated, but when these enzymes are dephosphorylated, they are inactive. Therefore, each of these will be dephosphorylated. However, the enzymes involved in promoting anabolic processes when dephosphorylated are active. However, the enzymes promoting catabolic processes when dephosphorylated are inactive. Glycogen synthase again promotes glycogenesis. Acetyl-CoA carboxylase promotes fatty acid synthesis. Phosphorylase promotes glycogenolysis. And triacylglycerol lipase promotes beta oxidation. We'll see that insulin is a key signaling hormone involved in regulating these anabolic processes in the fed state. So this diagram shows the metabolic effect and target enzyme of each of these processes, and this is representing the effect of insulin on each of these processes. So we see that insulin promotes increased glucose uptake in the muscle and the liver, and you see the key enzyme involved. It promotes glycogenesis in the liver and the muscle, of course, increasing glycogen synthase activity. It decreases glycogenolysis, therefore deactivating phosphorylase. It promotes glycolysis and also increases fatty acid synthesis. by increasing activity of acetyl-CoA carboxylase. We can see a diagram here of the effect of insulin on glucose and amino acid metabolism. We can see here as a summary that insulin stimulates the uptake of glucose, fatty acids, and amino acids by both the muscle and adipose tissue, and we see the overall effect of insulin binding to the insulin receptor, promoting the synthesis of proteins from amino acids, promotes the formation of glycogen, and reduces the breakdown of glycogen. It also promotes the synthesis of triacylglycerol molecules and increases glycolysis. Now we will review the hormones and other metabolic signals elevated in a fasting and starvation state. So we can see a number of these hormones or other metabolic signals that will be elevated, and then we'll see the effect of these hormones or other signals on key enzymes regulating anabolic and catabolic processes. So hormones and key signals that will be elevated again will be the AMP-activated protein kinase, glucagon, which increases in response to a reduction in blood glucose levels. We also see increase in epinephrine, norepinephrine, cyclic AMP, and glucocorticoids, and these will be elevated during exercise. We see that the result of these hormones and other metabolic signals is largely going to promote the phosphorylation of key enzymes, and some of the main key enzymes that we've been discussing have been glycogen synthase, acetyl-CoA carboxylase, phosphorylase, and triacylglycerol lipase. We'll see with the phosphorylation of these enzymes that the enzymes promoting anabolic processes will be inactivated, and those enzymes promoting more catabolic processes will be active. So activated will be the phosphorylase enzyme promoting glycogenolysis, or the breakdown of muscle and liver glycogen. Also, we'll see that triacylglycerol lipase, which will be promoting the breakdown of triacylglycerol in the adipocyte to the free fatty acids and the glycerol backbone. That process will also be activated by these hormones or metabolic signals. Now in the next 3 slides, we're going to review again the dominant processes and the hormone and signals that are going to be responsible for activating these processes during here the fed state. We'll also talk about the post-absorptive and early fasting state, and then the starvation state. So again, the dominant processes of the fed state will be glycogenesis. This is going to be activated by glycogen synthase. Also, we'll see protein synthesis, fatty acid synthesis, which should be activated by acetyl-CoA carboxylase. We'll also see lipoprotein and cholesterol formation. Here we'll see the key hormones and signals that will be elevated. However, insulin is going to be the main hormone or signal that's going to be involved in activating glycogen synthase and acetyl-CoA carboxylase. Next, we'll see the processes dominant in the post-absorptive early fasting state and the hormones or signals that will be elevated and responsible for activating these enzymes. So dominant in the early fasting state would be primarily glycogenolysis, or the breakdown of muscle and liver glycogen to free glucose and to be involved in increasing blood glucose levels. Also, we see early initiation of gluconeogenesis, and this is particularly going to be involved in the alanine glucose cycle versus the breakdown of muscle protein. And we also see some initiation of beta oxidation with the activity of triacylglycerol lipase. AMP kinase is going to be a key metabolic signal, increasing catabolic processes and decreasing anabolic processes. However, we see that glucagon is involved with promoting glycogenolysis of liver glycogen, and the catecholamines, particularly epinephrine, are involved in promoting glycogenolysis in the muscle. Lastly, we'll see the dominant processes in a more prolonged fasting or starvation state. The hormones and signals that will be elevated in this state are going to be similar to the early fasting state. However, we're going to see a shift from glycogenolysis and gluconeogenesis to the process of ketogenesis and beta oxidation being dominant during this more prolonged fasting and starvation state.